So here we go. We're going to start to talk about the reabsorption process. And so this begins, essentially, we've just now exited the glomerulus, and we're now into the proximal convoluted tubule. And remember that the majority of the fluid and contents are still out in the capillary. Some of them are in the tubule itself, and we'll talk about kind of where we have things and how much is there in just a moment. So in order to get reabsorption to occur, the majority of things are going to be transported passively. Water is going to move back through the membrane via osmosis. So there we have another passive process for this to occur until we basically reach an equilibrium, until we have essentially a process where both the capillary as well as the tubule are basically the same concentration, and the amount of osmosis and the amount of movement of the solutes is going to slow down the closer we get to that equilibrium point as well. Some of those processes themselves, though, have to be active, and so we have to expend some energy in order to get some of those substances back into the body. Things like glucose, amino acids, sodium, we're going to expend some energy in order to get those substances to come in because we want to make sure that we still have them back into the body itself. So when we look at this osmosis, and so the proximal convoluted tubule, and especially as we move down into the loop of Henle as well, we have some changes that are occurring along those loops. And so inside the tubule, we took out water. And so some water came out of our glomerulus when we first started. A large amount of that water is still in the capillary as well. So we've got water there. From there, we have other things that are still inside the capillary and the tubule. We have things like our sodium. And so we have sodium on the two sides as well. And so that amount of sodium that's going to be there is going to be slightly different between the two sides as far as our overall component that is there. And so we're going to have some sodium here on this side. We're also going to have some sodium over here. On that side as well. Change colors here. Go with the yellow. We're going to have some calcium. That is over here as well. Between those two sides, we'll have some potassium. That is there also. And then some of the other things that we're going to have are going to be things like our, let's go with this red, our red blood cells. So we have an RBC there. We're also going to have things like our white blood cells as well. Let's go with dark blue for our white blood cells. That is there as well, inside there. And then we're going to have things like even platelets. Out there inside the capillary as well. And then the last thing that we're going to have here, find one more color, is we're going to have proteins. And so we've got our plasma proteins over here as well. So we've got our plasma protein here also. And within that, we can now see that we have essentially a difference between the two sides as far as their concentrations go in terms of the amount of substance that are in each one. So this generates a process where the capillary is a hypertonic solution and the tubule is a hypotonic solution. And so we talked about solutions before, way back when we were talking about the cells. And with that, when we have a hypertonic solution versus a hypotonic solution, we're going to have the water wanting to move to the opposite side. And so the water is going to move towards that higher concentration, towards the hypertonic solution, and that water is going to move across passively and be able to move to the other side until we start to get a general change in the concentration between the two sides, to where we now have equal concentrations on those two sides within the two components that are there. And so our capillary and our tubule now have two different components to them. But as they move down along, and as we're moving through that tubule, we're pulling some of our other components that are there are going to be getting pulled out as well. I think I've run out of colors here for new colors. But the other things like our glucose, our sodium, which I had up there as red, just erase that one there. So our sodium is going to move through there also and make its way across on there, and our glucose is going across also. So all that is happening within that proximal convoluted tubule, and when that is occurring, we're having this occur at that epithelium. So we have the lumen of our tubule, we have the capillary itself, and in between those two, we have kind of two different spaces as well. We have the epithelial cell itself, and so of the tubule, and it has this kind of rhombus type of a looking color and things like that that's there. And then we have the actual interstitial space, so we have fluid that's there as well, and components are going to move typically through the cells, and some of the components, especially water, can slip through some of those tight junctions as well. And so the cells themselves are held nice and tightly together in most places via those tight junctions. And so we'll see the individual steps that we have to go through here coming up in the next couple of slides. So when it comes to these tight junctions, in a sense, not all tight junctions are created equal, or at least so much in their tightness, I guess you would say. And so as we go through the proximal convoluted tubule and down into the loop of Henle, the tightness, so to speak, changes. And so the components that are there are going to alter in terms of their ability to allow things like water to move through. And so the tight junctions in that proximal tubule, in terms of the proximal convoluted tubule, are essentially kind of looser, and so more water is going to be able to go through. As we move down through the loop of Henle, and we're getting further into the system in a sense, those tight junctions start to kind of tighten up, so to speak, and eventually becomes impermeable to water. And so we're decreasing the amount of water that can travel through and in between the cells, and so we're going to have a decrease in that component. So that's where, as we come through kind of the proximal convoluted tubules, down the loop of Henle, and then back up, going down, we're having water come out, and back up, the water is staying put, and it's the solutes that are going to come out. And so we're going to change kind of what's going to be able to be moving through those components, because now we're going to travel through the cells themselves. And so tubular reabsorption can be kind of broken down into 5 steps or 5 stages of things that kind of have to happen in order to get substances to move through the cells themselves. And so the first thing, and we'll look at the picture here in just a second, is they have to go through the membrane of our tubular cell. So they have to kind of cross that from the lumen through that first cell and make it through that membrane that's there. Then they have to go through the cytoplasm. Then they have to go through the membrane at the other end to get into the interstitial fluid. And then they have to go through the membrane of the capillary bed. And so kind of going through 5 different phases, in a sense, as it makes its way through there. And so the reabsorptive process itself begins here within the tubule, and we're going to start to move through our membrane and make its way through there. We're then going to move through the cytoplasm, make its way through the next membrane in that same cell. And then we're now number 4 here. We're into the interstitial fluid. And then from the interstitial fluid, we're going to move through the cell membrane of the capillaries and finally get back through. For sodium, we're going to have our sodium-potassium pump working to move that sodium in through the membrane and out into the space there in the interstitial fluid once again. And so the process itself is slightly different there. But the majority of substances that are going to be moved, we're going to potentially be able to do with a passive process, no energy involved. It can simply move down its concentration gradient. It can move across that membrane following that concentration gradient, going from higher concentration towards lower concentration always. The active process is going to require us to impart some energy, so it's going to take some ATP in order to get us to get those substances to cross through the membrane itself. And this process may be utilized only for some steps. And so just like with that sodium-potassium pump itself, there is some energy that's involved there, no matter what if all the rest of the steps themselves are passive. And so if any kind of energy is needed in any of the 5 steps to get across, so like for sodium there, in step 3, the sodium-potassium pump was utilized there, and so we had to impart energy. So that becomes an active process or an active transport across that membrane. Sodium reabsorption is one of our more important substances that we're going to move across, and when it comes to energy use, it's using up some 80% of all the energy that the kidney uses. And so active transport there in our 3rd step right in here to move sodium across the membrane is an active process. And so in general, when we think about kind of sodium and water, the two of them kind of follow one another. And so when sodium gets reabsorbed here, the water is going to follow with it. And so the water is going to move across that membrane as well. And so it's going to move across, and the sodium eventually ends up into the plasma of our membrane there, and with it is going to come the water. Or if we leave that sodium back out there, we're going to leave the water inside the lumen. So we're going to have water and sodium, and they're both going to stay in there. So kind of water and sodium follow one another. Wherever we move sodium, water is going to follow, and vice versa. That reabsorption not only is happening in the proximal convoluted tubule, but it's also happening down through that loop of Henle as well. And so in the processes as we move along, sodium is actively being pumped out of the ascending loop into the interstitial fluid. And so as we've moved down the loop, the descending loop, again, we have primarily water moving on the descending loop, and on the ascending loop, we primarily have solutes or sodium moving up through that membrane itself. And so we're going to increase the amount of sodium coming out into the interstitial fluid, and with that, that's going to draw water into there as well. And so that's going to pull the water along through as osmosis and get the water to move into those components in the interstitial fluid, and then eventually into the capillaries. And then next up, we'll start to talk about the process of secretion.